These are the notes for chapter 1.3 on membrane structure. When we were talking in chapter 1.2 about those really teeny tiny subcellular structures like organelles, like membranes, like DNA, it's really easy to take for granted our knowledge of such structures because they're really gosh darn small. And you know what? Electron microscopy is relatively new. So it's really amazing to think that long before we had these electron microscopes, okay, scientists were making models of very tiny cell structures that they couldn't see. And they were doing that based on their chemical properties and kind of figuring out how these pieces had to fit together. The two models of the cell membrane that you need to know are the Davison-Danieli model and the Singer-Nicholson model. You'll also hear this one referred to as the fluid mosaic model. So let's talk the Davison-Danieli model. Okay, I call this one kind of the sandwich model. Okay, because Davison and Danieli thought that the cell membrane kind of resembled a sandwich. They knew through chemical testing that the primary components of the cell membrane were phospholipids and proteins. So they use their best inference making skills and the limited data that they had to make a model that looks like this. To me, this looks like a sandwich. So they said, okay, well, we know it's made of phospholipids and we know it's made of proteins. So we think that the phospholipids are on the inside and on the outside, so facing out of the cell and inside the cell, okay, are our protein layers. So again, we're looking at a transect of the cell. Here's the outside, here would be the inside. Here's a transect or a cross-section of the cell membrane. So if you had to draw this, which, gee, I think you do, I would draw a couple layers of phospholipids. And I hope yours is better than mine, okay? And then kind of a layer of proteins like this, okay? And then probably I would label them. So these are the proteins. And then the phospholipids are in the middle. One of the ways that I remember this is the sandwich model, like D and D, and there's a dash in between it. So there's two equal things on the outsides with something in the middle. I don't know if that's helpful, just a little trick. So how did they come to this conclusion here? Okay, so they knew there had to be two layers just because of the way that the phospholipids interact with each other. We'll talk about that later. They also noticed that when they exposed the cell membrane to things that destroyed proteins, a lot of solutes were no longer able to pass in or out of the cell. So they identified the two main components, okay? They just had to do some more work with the rearranging of those proteins and phospholipids. And when they came up with this sandwich layer, the Davison-Danieli model, people were like, yeah, obviously, that makes perfect sense. And scientists and the rest of the scientific community really widely accepted this for over 20 years. If you look at some old school textbooks, this is what the cell membrane drawings look like. It's really cool to think about how this stuff is changing as we learn more. But the Davison-Danieli model and that little protein sandwich love story eventually came to an end. And that all happened with the invention of the electron microscope. Now, don't feel too bad for Davison and Danieli. This electron microscope kind of destroyed the assumptions and the works of lots of other scientists, okay? Because it really opened up our eyes and we were able to see things that we couldn't before. So when we looked at cell membranes underneath of an electron microscope, we were able to observe a couple key things. First of all, not all membranes were symmetrical. So that's interesting because here on the sandwich or Davison-Danieli model, it's clear that the top half is symmetrical to the bottom half. The half that faces outside of the cell is identical to the side that faces inside the cell. Well, that didn't really jive with some of the information we were getting from the electron microscopes, okay, that there were some inconsistencies there. The next thing that we notice is that membranes with different functions had different compositions. So different cells need to allow different things in and out of their cells. Your skin cells, for example, have a very different agenda in terms of letting things in and out than your nerve cells do. And we were looking at those membranes of different cells, we noticed that the structure or the composition was different, that the ratio of phospholipids to proteins wasn't always the same. Well, in this model, the ratio of proteins to phospholipids is always going to remain the same because we have a sandwich layer here and here. So that kind of wasn't going well. And then finally, a protein layer is not likely because it's nonpolar and doesn't interact well with water. So we said that here's the outside of the cell, here's the inside of the cell. Well, proteins, for the most part, don't interact well with water. They are what we call hydrophobic. They don't like water. So wait a minute. The primary component of our cellular fluids, the cytoplasm, is water. And most of the fluids that your cells are floating around in is made up of water. So it started to really not make sense to have these protein layers facing those water-filled environments. So when this stuff kind of came to light, a lot of people were looking for a new model of the cell membrane. And our friends Singer and Nicholson, they found it. Well, they didn't find it. They worked really hard and they tested a lot of things to come up with this crazy thing. We need to know it by a couple of different names. So it's called the fluid mosaic model, also known as the Singer-Nicholson model. And this fluid mosaic model really kind of helps us to understand way more about it than necessarily the people that named it. So when we talk about mosaics, those are things made out of many parts, just like a tile mosaic for my art friends. When we say fluid, it doesn't mean that you can drink it or that it forms the shape of its container. Fluid means things can move around. So I always say, like, the seats in my class have a very fluid situation. People can move around to their heart's content. They're not stuck in one place. So that was a really interesting proposition by Singer and Nicholson, okay, that we had these layers of phospholipids, but that these proteins that were stuck in them can move all over the place throughout the cell membrane. That was really interesting. So here's what we need to know about Singer-Nicholson. Okay, we're still talking about a double layer of phospholipids. However, instead of the layers of proteins like in the Davison-Danieli model, they thought that the proteins were embedded within the lipid bilayer. Okay, so these proteins, we can have different amounts of them in different cell membranes. We can have different types, and these proteins can actually move from spot to spot to spot. Okay, so what allows for all of those other irregularities that we were seeing with the electron microscopy. If you have to draw a picture, okay, that's going to look something more like this, where we have some phospholipids and then maybe kind of a protein or something sticking in there. Okay, looking like something like this. We don't need to go into too much detail because we're going to draw the fluid mosaic model and right about now. So what do we need to know about the structure of this fluid mosaic model before we draw it? Okay, well, it's important that you know how to identify parts in drawings that are given to you, how to draw them yourself, which we're going to practice here, and how to describe the functions. So right now we're going to focus on drawing and labeling. We'll go back and talk about the functions of these components in a couple minutes. Okay, so I always start with a couple phospholipids. I'm going to leave myself some room here on the side, okay, for my labels. because we all know that draw doesn't mean draw. Draw means draw and label. Okay, so here I've got 2 layers of phospholipids, okay, with their tails pointing inward and the heads pointing outside or inside of the cell. So I'm actually going to back up for a minute and kind of show you what's going on here. If here's my cell, okay, and then here's a really bad description of the cell membrane on the outside, okay, imagine what would happen if I kind of cut this cell and then I turned it over on its side. Okay, and when I do that, I'd be able to look at a cross section of the cell membrane. Okay, so here is going to be outside of the cell, and here is going to be inside of the cell. Okay, so again, looking at a cross section of the cell membrane. So phospholipids, here's the heads, here's the tails. Tails point inward towards each other. One of the things that we're going to see are proteins. Okay, so these are phospholipids. Okay, and this is what we call an integral protein. If you integrate into something, like, wow, you've really become a part of that something. So if you move to a new country and you really integrate into that culture, okay, that means you've done a good job of like assimilating, becoming part of that culture. And so an integral protein spans both sides of the cell. It goes from the outside to the inside. All right, so continuing on, I'm going to draw a few more phospholipids here. Okay, now one of the things that we're also going to find in our cell membranes are peripheral proteins. And peripheral proteins don't span the inside. In fact, this would be kind of covered up here with phospholipids. Okay, so they are only on the periphery, the outer edge of the cell. A lot of times these can be used for cell recognition, cell-to-cell binding, those kinds of things. Anchor points for extracellular matrix, etc. Alrighty, so I'm going to continue on here. Some more phospholipids. Okay. I can also have special types of peripheral proteins that have these carbohydrate chains on them. And this is called a glycoprotein. Okay, glyco meaning sugar. Okay, so here's my glycoprotein. And it's important to note that this is a sugar chain. So if you need to make a note of that, that's fine. I'm just running out of room here. Okay, glyco, okay, meaning sugar, protein here. We use these a lot for cell recognition. These are kind of like name tags for the cell. Every cell is going to have a different pattern of these so that they can recognize each other, especially important in the immune system. Okay, it's important to know if those cells belong in our body or if they don't. Okay, so here's some more phospholipids. Great. Now, if this is an animal cell, we're also going to find in this region here with the tails, and I shouldn't have, wow, that was really bad. I shouldn't have drawn it with sketch lines like that. I'm just going to draw a blob, okay? This is a molecule of cholesterol. So we often hear about cholesterol, especially if you watch a channel that a bunch of old people watch. There's about 1 billion cholesterol commercials and medicine that can help you lower it. Cholesterol is actually found in the cell membranes of our cells, and all animals have cholesterol in their cell membranes. We'll talk about why in a little bit, but it's important that you draw it. It's important that you draw it in this tail section, not up here with the heads, but kind of in the middle here with these tails, okay, because that's where it hangs out. We need to know its correct placement. One more thing I kind of want to label here. This is what we call the lipid bilayer. Okay, lipid because these are phospholipids. They're made of fatty acids. Bi meaning two layers of lipids. You may also want to label the hydrophilic, well, it would be helpful if I spelled that right, hydrophilic region. Okay, these round things, the heads of the phospholipids, are hydrophilic, water loving. And this section in here with the tails is hydrophobic, not so fond of water. Okay, water hating. We'll talk more about why it has those properties in a couple minutes. Okay, so I kind of want to return to this idea of phospholipids. Phospholipids are exactly what they sound like. Okay, they're a type of lipid with a phosphate group, and they have two ends. Hello, two, not two. Two ends. So we have the hydrophilic head, okay, hydrophilic, water-loving, there's the head, and the hydrophobic tail, okay? You may also hear those referred to as polar and non-polar. So polar and hydrophilic mean the same thing. Hydrophobic and non-polar mean the same thing. So when you're doing your drawings here, it's a good idea to always have both hydrophilic and polar. But a circle, two tails. So what is so cool about the way that our cell membranes is arranged? It's all because these tails hate water so much. So kind of like these people hate water so much, they're all sticking together under the same umbrella. These non-polar hydrophobic tails hate water so much that they will point inwards towards each other to stay away from the watery solutions that are inside and outside of the cell. So there's such a strong hydrophobic interaction there, okay, that it holds the membrane together. There is nothing physically attaching one phospholipid to the next. It's all because they hate this water so much that they're attracted to each other in order to stay away from that water. Okay, so let's chat about this molecule of cholesterol that we included in with our cell drawings. See, your membranes have a really cool property of being fluid, okay? Things being able to move around and being flexible. Sometimes the volume of the cell changes, okay? Maybe it's got more or less water in it. Sometimes we need to temporarily open up holes in the cell membrane. Sometimes our cell membrane needs to pinch into two during cell division. It has to do a lot of really crazy things, and it has to be able to do those things under a pretty wide range of temperatures. Okay, now, since the membrane is made up of lipids, okay, we can assume that these lipids act similar to the way the other lipids act when you expose them to a variety of temperatures. Well, ain't no one got time for all those crazy changes. So animal cells have adapted the ability to insert cholesterol molecules into this hydrophobic region of the cell membrane. And the cholesterol will actually keep this membrane more stable over a wider variety of temperatures. So that's the function of the cholesterol, okay? It's, again, in the hydrophobic tail region, helps the membrane remain stable over a wider range of temperatures. Plant cells don't have cholesterol in their plasma membranes because they have cell walls to help maintain shape and things like that. So, animals need cholesterol, but we don't need to eat it. See, our bodies can make every bit of cholesterol that we need in order to maintain these cell membranes. If you have a diet high in saturated fatty acids and cholesterol, sometimes that can lead to an increase or an overload of cholesterol in the bloodstream, causing all kinds of naughty things to happen, like heart attacks and other things. Okay, so we don't want that. If you're looking to cut down on your cholesterol, you should be eating plants because they don't have cholesterol in their membranes. Okay, so another component of the cell membrane are the proteins. We've got two major types, integral proteins and peripheral proteins. And that just tells us, do they span one side of the cell membrane or both? All right, so let's talk integral proteins first. Integral proteins have a really cool property called amphipathic. Okay, amphi, like amphibians. Amphibians spend part of their life on water and part of their life on the land. So we're talking about both, the ability to live in both environments. And what that means is that they have to get along with both the hydrophilic regions of the phospholipid bilayer and the hydrophobic regions. So they have to have parts of them that are also hydrophilic so they can get along with these guys, and they have to have parts of them that are hydrophobic so they can get along with these tails that are in here. Now, that's a lot different than the peripheral proteins. Peripheral proteins, that is really hard to say, peripheral proteins, they hang out on the edges, okay, the periphery, peri meaning around. And they don't really dig in too much to this hydrophobic region. They kind of just stay on one side or the other. Many of these peripheral proteins are those glycoproteins that we talked about. So here's a better picture of those glycoproteins, okay? Proteins and then the glyco or sugar chain. And we need to know that glycoproteins have a carbohydrate. That's a type of sugar. Well, that's not true. Sugar is a type of carbohydrate. But anywho, a carbohydrate or sugar chain attached to them. And this is really important with cell recognition. So it's one of the tools that our cells use to recognize each other. So if one of your immune cells bumps into one of your body cells that's supposed to be there, this one can't really say to this one, hey, do you belong here? What it does is it will sense these glycoproteins and it'll say, okay, your glycoproteins match what I've been finding in the entire rest of the body, so you must belong here. Excellent job, you can stay. If it doesn't belong here, if it doesn't have a glycoprotein that's consistent with the rest of the body, then our immune system starts to kick in because it thinks that it's an invader and it wants it out. So really important for cell recognition. Make sure you've underlined or make sure you've paid close attention to that part. Now, these membrane proteins, and again, it doesn't matter whether I'm talking about peripheral or integral, they have a total of 6 functions that you need to know. So there are 6 different things that proteins that are embedded within the membrane can do for our cells. There are a ton of different mnemonics out there. The one that I use is jet rat. Okay, so they can function as junctions. So they can literally connect cells together. They can function as enzymes. So proteins that are embedded in the cell membrane can facilitate cell reactions. The big one we're going to talk about is for transport, moving molecules into or out of the cells. Recognition, which we talked about, being able to recognize one cell for another. Anchorage points. Okay, we talked about the extracellular matrix in chapter 1.2. Well, the only way that an extracellular matrix can stabilize a cell is if the cell has a way to connect to that extracellular matrix, and it does it using their proteins. And then we have transduction, which really means just receptors for hormones. Okay, so hormone binding. If it were me, I would rather this say hormone binding than transduction, but I can't identify what RAH would be. That's not a real word. So if you have another mnemonic that you like to use, fine. Just make sure you know this is for hormones. One of the themes that we're going to see all throughout biology is the theme of form and function. Okay, so because these proteins have different functions, they're also going to have different shapes. Okay, so I'm thinking about shoes. Okay, shoes are kind of like proteins. They have different shapes for different functions, and so do these membrane proteins. Okay, so the different functions kind of correspond to these different shapes that we're seeing here. So all these cool properties, or cool structures rather, of the membrane kind of lead to some really awesome properties of the cell membrane. So first of all, it's very flexible. It can move and form a variety of shapes. That's super important. Okay, maybe it's got to squeeze through something, or maybe it has to expand. They're also very strong. This hydrophobic region, these tails hate water so gosh darn much that they will cling to each other as to not let water in and touch them. So it's a very strong membrane. It's what we call self-healing. We poke holes in these membranes all the time. We'll learn more about that in the next chapter, but sometimes in order to move things in or out, okay, we have to poke a big hole into it. So I'm thinking of like surgery. Sometimes if you need to get something out of someone's body, you gotta poke a big hole in it and rip it out. Cells aren't all that much different, okay? The only difference is they don't have like Dr. House to sew them back up again. So they're what we call self-healing. Okay, that membrane will close any gaps by itself. Again, that's because these hydrophobic tails hate water so much that they would rather cling together than let water in and touch them. So any gaps will kind of reform together. And then they're what we call semi-permeable. Semi meaning partly, permeable meaning able to get through. Only some things can get in and out of the membrane. Okay, we're going to talk a lot about that in the next chapter, but it's important to kind of keep this word in the back of your head for now. And that's it on chapter 1.3 on membrane structure.